Magandang hapon po sa lahat. Ako po si Dino Tordesillas at bago po magsimula ang lahat ng kaganapan ngayong hapon, nais ko pong imbitahin na lahat para sa pag-awit ng pambansang awit ng Pilipinas.
Mga kababayan, ang pambansang awit ng Pilipinas.
Kayang magiliw, telas ng sinakalan, alam ng puso, sa dikit mo'y buhay. Lupang hinirang, tuyag ka ng magiting, sa manlulupin, di ka pasisingil, sa nagagang tutok sa simoy at sa langit mong bagaw, ay kinatang tula, kawin sa pagkayang minamahal, ang isang ng matawad mo'y tagumpay na nag-iingin, ang bituin at araw niya kailan pa may di magtitingin. Lumang ang araw ng walhagin pagsinta, buhay langit sa piling mo. Ang inigaya ng pagkay mga aking, ang mamatay ng dahil sa'yo.
Again, a good afternoon, a good day to everyone. I am Dino Tordesillas. I am the current Secretary of the Philippine Society for Developmental Biology Incorporated, and I am very happy and excited to welcome each and every one of you in the first in our webinar series for 2022 in Animal and Plant Developmental Biology. This webinar series is presented to you in cooperation with Macare Medicals Incorporated and our sponsors, Merck, PhilAnaserve, and MedTest. Okay. So again, this is just the first of two sessions that we have for this webinar series. So today, 23rd of April 2022, we have two speakers who will be talking to us about the use of nanobiosensors for agricultural applications, and also how we can enhance livestock production through research and reproduction research and innovations. Next week, we will have two more speakers for session 2 of this webinar series. Before we start, I would like to make sure that the rules are clear for this webinar, as we do here in PSDB. So we would greatly appreciate it if everyone will turn off their audio and video upon login and while the speakers are presenting their or talking about their research. We do have a specialized app for questions later on. So we do have this Slido, which is an external application, but this will really help us organize your questions. So you can scan this QR code, and our other webinar moderators will be posting the link to this to Slido time and again in our chat box. So please just backread, or you can right there now. So you have a link to the Slido. So they will be posting it every now and then. So if you do have questions, go to Slido through the link or through this QR that you are seeing right now, and you can type your questions there. For those who are PSDB members, links to the evaluation form will be posted inside the chat box at the end of the session. So later, after our second speaker, we will post the link to the evaluation form for this session. Okay, the first session in our webinar series. Okay. How do you get the certificate for your attendance and your participation in our webinar series? You must be an official member of PSDB, and you should have registered in this PSDB 2022 webinar series. You should also submit your evaluation form for each session. So, yeah, I believe we will be sending out evaluation forms for the 2 sessions. And please remember that the evaluation forms will open at the end of the session. Okay, so after posting, we will activate the evaluation forms after our second speaker, once we post the links later on, until only 11:59 p.m. today. Okay, so that's the 23rd of April, 2022. So the evaluation link will only be active, or the evaluation form will only be accessible just before midnight tonight. So please take note of these reminders. If you do want, or if you need a certificate of participation in our webinar series. Okay. Now, before we proceed to our speakers, I will be presenting or sharing to everyone the President's message, of course, delivered by our PSDB President, Dr. Larry Ann Ipulan Colette. We just share her recorded message.
To our active members, distinguished speakers, and PSDB webinar attendees, good afternoon, and welcome to the second PSDB 2022 webinar series. It has been a tough 2 years for everyone, both professionally and personally. Many changes in education and research dawned upon us during the pandemic. While there is limitation in our mobility, PSDB believes that learning and improvement can happen. Hence, the birth of webinar series during the early half of the year, which is separate from our annual convention held during November. We are proud to have our new set of esteemed speakers for this webinar series. Today, session 1, our speakers will discuss innovations and advancement in Philippine agricultural studies and animal reproduction. For session 2, that is next Saturday, our speakers will talk on floral stem cell fate and monitoring of endocrine disruptors. All these studies use developmental biology concepts to progress in their respective fields. We are thankful for their expertise and perseverance for furthering research in the field of developmental biology. We hope that you will share their enthusiasm in discovery and learning as we listen to their talks. Mabuhay kayo na patuloy na nagpupunyagi para sa pansarili at pambansang pag-unlad. Maraming salamat sa inyong suporta sa mga activities ng PSDB.
Okay, that was the message from our dear President, Dr. Larry Ipulan Colette. Let me go back again to our slideshow. So there, again, thank you very much for your message, Dr. Larry Ann Ipulan Colette, PhD, our PSDB President. So, okay. So for our first speaker, our first speaker for this afternoon is Dr. Lilia M. Fernando. She's an associate professor in this Institute of Crop Science, College of Agriculture and Food Science in the University of Philippines, Los Baños. Dr. Fernando is a biochemist whose research is on the utilization of secondary metabolites from plant growth. from plant growth promoting bacteria for plant production and propagation. She works on the synthesis, characterization, and utilization of biocompatible nanomaterials for the improvement of crop productivity, along with safety studies of nanomaterials in plants and microorganisms. Her research team also develops nanobiosensors for agricultural, environmental, and food safety applications and medical diagnostics. Her team has filed for patents and published journal papers from their research and collaborative endeavors. As I've said, Dr. Fernando is an associate professor at the Institute of Crop Science at the University of the Philippines, Los Baños, where she teaches agricultural biotechnology courses. She is also an affiliate professor of the Institute of Chemistry of the UPLB, where she teaches biochemistry courses. She is the co-founder of Makiling Tech, a university spin-off, which aims to facilitate the commercialization of microbial inoculants and nanobiotechnologies developed by their team through the Department of Science and Technology and other granting agencies. She obtained her PhD in biochemistry, Masters of Science in Biochemistry, and Bachelor of Science in Chemistry from the University of the Philippines, Los Baños. She is a DSD scholar and was recipient of the Overseas Sandwich Program, where she conducted her PhD research in Michigan State University. She had her postdoc studies at the Massachusetts Institute of Technology, where she worked on phage and yeast display techniques for agricultural applications. So here to talk to us about nanobiosensors for agricultural applications, we have Dr. Lilia M. Fernando.
Thank you, Sir Dino, for the kind introduction. I hope my audio is fine. Okay.
Yes, ma'am, loud and clear.
Okay, thank you. So let me share screen. Okay, so good afternoon, everyone. And before I start, I'd like to thank the Philippine Society for Developmental Biology for allowing me to share our research on nanobiosensors for agricultural applications. Okay, so as I mentioned earlier by Sir Dino, our team focuses on nanotechnology, which is the utilization of particles between 1 to 1,000 nanometers. However, there's a strict definition now on nanotechnology where you're required not to utilize 1 to 100 nanometers. Well, it depends on the application. So this slide shows the scale, showing the dimensions or the scale for the dimension of different nanometric and micrometric objects. So we have here the dimension for plant and animal cells, so 1 micrometer to 100 micrometers. And then for viruses, about 100 nanometers, probably 50 to 100 nanometers. And then proteins, about 10 nanometers. And we have DNA, 1 to 10 nanometers. And for atoms, 0.1 to roughly 1 nanometer. So for nanotechnology, we focus on this dimension, specifically on the application that we're working on for nanobiosensors. Now, when we talk about nanotechnology, we have several nanomaterials that are utilized. We can use nanocapsules, metallic nanoparticles, nanowires, nanotubes. So our team focuses on nanocapsules and metallic nanoparticles. But for my talk today, I'll be focusing on the utilization of metallic nanoparticles for nanobiosensors. In relation to developmental biology, well, our research, specifically the one that I'll be sharing this afternoon, probably will help sa developmental biology as a tool or technique in other research or sa mga discovery pa natin in biology. Okay, so as mentioned earlier, my research focused on plant growth promoting bacteria, and we also dealt with its application we harness nanotechnology for the improvement of crop productivity. So this is our research framework for the use of nanostructured materials for crop improvement and protection. And the one that I'll be sharing this afternoon will focus on nanosensors or diagnostics. So our work on nanobiosensors started through our collaboration with DOST Balik Scientist Awardee, Dr. Banji Alusilha. She was actually my host when I had my PhD dissertation conducted at MSU. Okay, so this is the platform that we're using for the nanobiosensor. So we have the analyte. Usually the analyte that we use is the DNA. So we have the DNA-based nanobiosensor in the lab. So for instance, for COVID, if we have COVID virus as our analyte, we have to extract the virus and have a bioreceptor, which is usually an oligoprobe, okay, because we're dealing with DNA. And the transducer that we're using is the nanoparticle. It's the gold nanoparticle, and sometimes we make use of the iron oxide or the magnetic nanoparticle to concentrate our sample. And we use an electrochemical method. We make use of a potentiostat, a battery-operated potentiostat in our nanobiosensor design. And just like a pH meter, we use a screen-printed carbon electrode. The size like our thumb lang, maliit lang siya. And the idea is for us to bring this battery-operated sensor or device in the field for detection of various pathogens. So why the need to use nanobiosensors? Nanobiosensors, or the use of nanobiosensors, will result to rapid detection time, higher sensitivity and specificity, and it has compatibility with data processing technologies. This is a table showing the difference among different methods of detection. We have 1, 2, 3, 4 detection methods here. We have plating, immunological, nucleic acid-based technique. This can be the PCR technique, and nanotechnology-based techniques. And if we look at the parameters identified here, the nanotechnology-based techniques has higher sensitivity or have higher sensitivity compared to the other methods. And also the duration of the assay is shorter compared to plating, PCR, and other techniques. So this is our nanobiosensor program. We started with the detection of the following pathogens: E. coli, Listeria, Salmonella, and then we dealt with plant diseases. Then we collaborated with De La Salle and St. Luke's for the detection of tropical diseases like dengue and JEV, or Japanese encephalitis virus, but we focused on biosurveillance. We detected the following viruses in mosquito. And well, this is just a plan to work with animal and veterinary science for the detection of the following contaminants: viral, bacterial, and chemical contaminants, and also organic fertilizer. And we also talked with the DOH for the possibility of collaborating for the design of for the design of nanobiosensors for the detection of bioterrorism agents. Well, I think this webinar can be a venue as well for our team to collaborate with you in terms of the application of nanobiosensors. So my talk will focus on the application of these nanobiosensors or these devices in the detection of plant diseases. The FAO estimates that annually between 20 to 40% of global crop production are lost to pests. Each year, plant diseases cost the global economy around 220 billion and invasive insects around 70 billion. So malaki po ang loss ng ating mga farmer. Aside from our current problem, if we hear from the news na hindi nabibili, overproduction yung iba nating mga produce like onions, but still our farmers struggle with pests. and we need to do something on that. So with increased trade and travel, the risk of plant pest spreading to new areas across borders is now higher than ever before. Each day, we witness a shocking number of threats to the well-being of our plants and by extension to our health, environment, and economy. So dito na po pumapasok yung idea din natin on One Health. So the term is biosurveillance. Ito po siguro yung kailangan ding maging focus ng ating bansa, no? Kasi minsan kung kailan nandyan na, saka pa lang tayo gumagawa ng solution. What if we have a proactive biosurveillance team, not just human health, but even in our agricultural produce, no? Malaking bagay po to, not just in plants but also in livestock. Madami po tayong challenges. And I'm glad that the Department of Agriculture supports as well our the biosurveillance, the rationale of the biosurveillance system po para sa ating country. Okay, so our work on the detection of plant pathogens was first applied in cacao. So yung collaboration po namin was with Dr. Divina Amalin of De La Salle University, along with our balik scientist, Dr. Vanji Alusilha from MSU. So cacao, lahat tayo, karamihan siguro favorite ang chocolate. So cacao is a commercially important plant species grown primarily for its beans. One of its many diseases, called black pod rot, accounts for up to 70% to 80% of the annual cacao production loss. So we have one MS student who first worked on a DNA-based electrochemical nanobiosensor for the detection of Phytophthora palmivora. So Phytophthora palmivora is the causative agent for the black pod rot. So ganito po yung symptoms ng mga cacao pods infected with Phytophthora palmivora. So they're not marketable anymore. So kawawa yung ating mga cacao farmers. Now, the conventional detection methods for Phytophthora are the following: culture method, PCR, and ELISA. So medyo mahirap po ito sa farmers. So when we talk with farmers in Isabela, sabi nila sana before daw magtanim, we can do something, because most of the pathogens infecting plants are soil-borne. So what if we have a simple sensor na pwede nilang gamitin sa mga farms nila, so they'll know if they can plant cacao already. Baka kailangan mo nilang magkaroon ng mga pesticide application. I'm not pertaining to chemical, kasi we can work on biological control agents as well before they plant their crop of interest. Okay, so our technique has two steps. Yung pinakauna po, is the triage or the screening technique. So ano po bang ginagawa dito? We have our sample. For instance, we get a swab from the pod of cacao and lagay namin sa microfuge tube. Then we add magnetic nanoparticles. So these modified magnetic particles can give us an idea if the sample has pathogen or none. So ang nangyayari po, nagkakaroon ng matting or spreading of the nanoparticles when there's pathogen. So this is not a specific detection yet, but it can help. Pag marami po tayong samples, we can screen kung ano lang yung gagamitan natin ng further diagnosis. So with matting or spreading of nanoparticles, there's pathogen, but pag walang spreading, just like this tube, negative po siya. So that's just our simple procedure. And from that, we proceed with the DNA-based detection. So lahat po na nag-positive dito, we will be identifying, kung yun ba ay Phytophthora or other pathogens. It's dependent now on the oligoprobe that we use. So eventually, no, yung plan po, we can have, by surveillance system, we can have a database na hopefully makonect natin ano, sa iba-ibang agencies natin or regulatory agencies natin dito sa country. Kasi ang problem minsan, yung mga halaman natin na may sakit sa, halimbawa lang po sa Mindanao, nadadala na sa Luzon, na hindi tayo aware, hindi aware yung farmers natin na yung nakahawa na pala yung kanilang mga in-order na seedling. So we should give attention ano, to that, to those concerns ng ating mga farmers. So as I mentioned earlier, this is our triage technique. And actually po lahat ng to, nag-start yung proof of concept sa medical application nitong nanobiosensors. This was first used in the detection of tuberculosis and dengue virus. So clinical samples to. Then through our discussions with our consultant, sabi niya, why not use it to plant test? Tinignan po namin, pwede pala. That's why we started working on the application in agriculture. So this image shows the positive detection of Phytophthora culture in our tube. So aside from Phytophthora, we also worked on Lasiodiplodia. Ito naman po yung nagkakos ng vascular streak disease. Talagang mamamatay yung buong cacao tree, yung buong halaman. Pag infected na po nung Lasiodiplodia, systemic po yung kanyang effect, no, sa halaman. So for this step, the triage technique, we're currently working on a method on how we can quantify the area nor the spreading of the magnetic nanoparticles. So for now, we're using image J, and hindi pa po tapos yung aming optimization since we're still working on actual pure cultures. Our ongoing project is on the validation of this proof of concept technologies. Now, I'll proceed to an overview of the DNA-based diagnosis. So we have two. Ito po yung aming dream, magkaroon kami ng kit for the detection of plant pathogens on-site detection of plant pathogens. So we can have a colorimetric method or an electrochemical method. So electrochemical is more sensitive. So ang ating magiging mga adapters siguro for colorimetric will be our farmers, our cooperatives, and for the electrochemical, some laboratories, private laboratories can make use of this more sensitive method. So this is the potentiostat that we have. It's battery operated. Halos kasing size na lang po siya ng mga smartphones natin. And this is the screen printed carbon electrode. The circle part is where we drop about 50 to 100 microliters of the DNA that we've extracted. So yung challenge po dito is our extraction, on-site extraction of the DNA. We're working on some techniques now para hindi tayo gagamit ng centrifuge and other equipment na wala sa field. So slowly po we're working on that para talagang on-site na yung maging detection natin for pathogens. The actual detection method, itong setup po na to, less than 1 minute lang. Ang matagal lang talaga ay yung ating preparation ng samples. Okay, so just to give you an idea on one of the platforms that we're using for the DNA-based detection, we have here a magnetic nanoparticle. So this black circle, these two black circles represent magnetic nanoparticle, and this is oligoprobe 1. Now we have another probe. The blue tail here is probe 2, oligoprobe 2, and we make use of biotin. So later on, we'll know why we use biotin. Halimbawa, ito po yung ating target DNA. So through Watson and Crick-based pairing, because our oligoprobe should be complementary po dun sa sequence ng ating target DNA. So there will be hybridization of the oligoprobes and the target DNA. Now, by virtue of the magnetic nanoparticle, there will be capture, magnetic capture. We make use of a magnet to capture the hybridized target DNA dun sa sample. So this can be replacement pag nasa field sa centrifugation. So once we have the sample captured, we embed it in a screen printed carbon electrode where we have gold nanoparticles just to amplify the electrochemical signal. And yung VPO dyan, this one is streptavidin. That's why there's a need for biotin, because there's a special affinity between biotin and streptavidin. And after that, we go to electrochemical detection. So again, this is just one of the platforms that we use in the electrochemical nanobiosensor. So optimizations required include the amount of magnetic nanoparticles used, and even the concentrations of the DNA. So this figure just shows that there's an optimum amount lang po ng DNA that we can use for the nanobiosensor to be to be able to detect our sample. Sometimes there's an overcrowding. We just use the term overcrowding in the lab. Pag masyado ng maraming sample, then namamask yung electrochemical signal. Pag masyado pong maraming sample. So this will be part of our of the methodology that we'll be recommending. So in terms of selectivity, just to show you, we used several plant pathogens. So this is for the DNA-based sensor for Phytophthora palmivora. So we used other plant pathogens like Lasiodiplodia, Coleotrichum, and Fusarium. So indeed, we have a higher selectivity for Phytophthora palmivora using the DNA-based sensor fabricated for this pathogen. And the limit of detection, so we can detect as low as 0.1 nanograms per microliter of the DNA. So sorry po, medyo malabo, but again, we want to have this device, the potentiostat attached to our smartphones, and eventually the data collected be available in the cloud to be shared to regulatory agencies and other stakeholders who will be needing this data. So in relation to the cacao roadmap, our team shares with the Department of Science and Technology in their objective to promote cacao, and eventually makatulong po tayo para dumami pa yung ating mga cacao farmers. And they'll be using some of the technologies that were generated through our DOST funds. So now we're in the validation and pilot testing of these nanobiosensors. And this is done by one of our students, Miss Marinold Purificacion. So she focused now on Lasiodiplodia, which is the causative agent for the vascular streak disease of cacao. So, ganun din po, we make use of the triage technique. So, just to show you how we do yung triage technique na pinakita kanina, yung spreading or matting of the nanoparticles, we have our buffer, and then we add the spore suspension or our analyte, then we add the magnetic nanoparticle, we mix and incubate at room temperature, palang mga lang po to, mga 10 minutes. And then we remove the supernatant. We actually use here a magnet, a magnetic rack, para ma-pull natin yung magnetic nanoparticles. So after removal of the supernatant, what's left is the magnetic nanoparticle with the pathogen. So we can make use of this kapag nag-spread siya or nagkaroon ng matting, we can use this in succeeding diagnosis. So as I mentioned, we make use of image J for now for the quantification. So same as I mentioned earlier, we do DNA-based detection, but for latiodiplodia, I'll just give you an example on the colorimetric detection that we had. Because earlier I showed about the results of the electrochemical detection. So the mechanism for the colorimetric detection is shown in this slide. So we use gold nanoparticles, thiolated gold nanoparticles. So the red circles represent the gold nanoparticles, and these threads here are the oligoprobes. Now, if there's a complementary target or our desired target, or yung analyte natin is present, the nanoparticles will be separated. So hindi sila nagka-clamp, because there's the target, complementary target, which hybridized with the oligoprobe will provide spacing para magkaroon ng distance dun sa mga particles natin. Hence, the solution will remain wine red. So our gold nanoparticles, wine red. So wala pong aggregation. However, if there's non-complementary target, then nagkakaroon po ng aggregation. So the solution turns blue to violet from pink or wine red. So another slide showing the same mechanism. So we have the tube with the target DNA. We add the gold nanoparticle probe with the oligoprobe with the gold nanoparticle. Then we add sodium chloride. Then it remains red if we have the target DNA here. But if we have the non-target, nagiging purple siya. So ito po yung magiging difference sa color. Pink to purple if we have the non-target, or if it's the negative, negative yung result. So these are some parameters that we optimized for the development of the nanobiosensor. Okay, so again, showing you the colorimetric detection of L-thiobromate. So this is the blank. Although dito may siya, medyo nag-pink siya. This is another, it's the same mechanism as I've shown earlier. So nag-retain po siya na medyo lighter ito, yung sa blank natin. I think the gold nanoparticles used were not the same. Okay, so this is the blank, and this is with this is with L-thiobromate, and this is with Phytophthora palmivora and L-thiobromate plus Phytophthora palmivora. I think there's a different mechanism here that we use. Hindi ko napakita po yung slide, but again, we make use of differences in color. So for this particular detection method, if we want to use L-thiobromate oligoprobes in this method, so dapat ang makuha natin na mag-positive yung L-thiobromate. So dapat mag-pink siya. So we just compared the two using L-thiobromate and Phytophthora palmivora. So negative po yung Phytophthora palmivora dito, clear yung tubes or yung solution, because the oligoprobe that we used for the detection is for L-thiobromate. So malaman ng farmer if there's latiodiplodia if the solution turns pink. Okay, so this is a combination of the two pathogens. So just to show you the signals in terms of absorbance readings from the detection. So L-thiobromate has a lower absorbance reading because it remained pink. Actually po, these wavelengths correspond to the absorption wavelength for the pink, color pink ng ating nanoparticles. So in terms of applications pa in other pathogens, we're working on the detection of other positive agents po ng Panama disease like Fusarium, Banshee top. So these two are for banana. In white potato, we have the following. In tomato, bacterial wilt caused by Ralstonia. In mung bean and peanut. So we make use of the same platform, the triage and the DNA-based detection. So this is with banana banshee top virus. These tubes are positive with BBTV, and this is for Fusarium. These tubes were positive for Fusarium, and these were the black. And this is for Ralstonia. Again, these are the positive tubes. And now where we have the cultures of Ralstonia added in the tube with the magnetic nanoparticles. And for the biosurveillance or the DNA-based detection, again, we have a colorimetric method used here. So for the detection of Ralstonia solanacearum, in this case, we first used synthetic DNA. This was done during the pandemic. We're not able to go to the field, but luckily we were able to order synthetic DNA, corresponding to the target sequence in Ralstonia. So the left image or image A is for Ralstonia, and is the non-target. So again, it turns pink when we have Ralstonia in our sample. Still, we're working on some optimizations in this methodology along with our validation studies. Now, in addition to those, we're now working on some post-harvest applications of the nanobiosensors. We have a project now with DLSU and funded by the OSTP card on the detection of the causative agents. Or for the, well, we want to focus on the microorganism producing toxins like aflatoxins, ochratoxin, na nakakababa po dun sa marketability ng ating mga processed na cacao beans. So again, this is the dream of our team to have this kit available to our farmers and stakeholders. So this is the nanobiosensor design and our applications. These are technology roadmap. Well, we filed for IP protection and, well, it's not efficacy trial, but more on validation studies. So now we're into pilot testing, and hopefully, we can do further validation and have the product registered. Medyo still a long way for us, but hopefully we'll get there, through perseverance pa rin, despite the challenges. And eventually, ma-transfer po natin yung ating technology to our beneficiaries. So our team hopes to address the following SDGs: good health and well-being, clean water and sanitation for our food and waterborne for our nanobiosensors for food and waterborne pathogens, and responsible consumption and production. On behalf of our team, I would like to thank you. And again, our team is open for collaboration and even for students who would like to work with us. Thank you and have a great afternoon.
Maraming salamat po, Dr. Lilia Fernando. At I never thought na ganun pala komplikado yung, I mean, this is chocolate we're talking about, and I never thought that it involved a lot of technology when it comes to checking the quality and making sure that our chocolate or cacao products are of the highest quality. Okay? So salamat po ulit, Dr. Lilia. So okay. Okay. So for this at this point, okay, we will have our open forum. This is open forum number 1 with Dr. Lilia Fernando. So this is the QR code for our Slido. the link is also in our chat box. So if you do have questions, kindly send them through Slido with the following format: your name, and also please include your affiliation and your question. And to facilitate the open forum for this afternoon, we have Mr. Jodel O. Galapon, our PSDB, one of our PSDB board members. Take it away, Sir Jodel.
Thank you, Sir Dino. Hello, Dr. Rinig po, ma'am.
Yes. Hello. Good afternoon, Sir Jodel.
Good afternoon, ma'am. Thank you for that very informative presentation for our participants and also for our students that are watching now in our YouTube live platform. So today we will be using this Slido to crowdsource all the questions from you, and then here are the questions, Dr. Lilia, from our Vice President, Dr. Nadia Abisimas. Her question is, can nanobiosensors also be used to detect the entry of pathogens from imported crops, livestock, or meat into the country? Are there countries that do this?
Okay. Yes, thank you for the question, Ma'am Nadia. Yes, we can apply the nanobiosensors for that purpose. Actually, ano nga as I mentioned, we should be proactive kasi marami talagang diseases ngayon na hindi natin nakapasok na sa country natin, hindi pa natin alam. And kapag malala na, siguro yung ating next speaker, who's into animal science can relate to that because we still have problems yata sa ating sa swine industry. So yes, plants po, malaking application yon. Hopefully we can have the BPI, we can have the Bureau of Plant Industry. Tingin ko sila yung agency who's involved in the regulation. Hopefully, they can make use of this technology sa ating mga borders. Pag may mga dalang halaman yung mga papasok, we can have a simple procedure there para malaman agad if positive siya or negative for a plant pathogen. So yes, there are exciting opportunities for this research. Thank you, Sir Jodel.
Another question from our board member. Our president po, Dr. Larry Ann Colette. Some basic questions, ma'am. Why spreading or mating happens during detection? What is the % accuracy of mating?
Okay. Yeah, that's a good question. Actually, we can't explain yet. Our collaboration with Michigan State involves the use of these nanoparticles. They're the ones supplying us with the magnetic nanoparticles, and they modified the surface of the magnetic nanoparticles such that the pathogens will stick on the surface of the nanoparticle. So, tingin ko lang po yung principle niya. They modified the nanoparticle such that yung tingin ko may mga carbohydrates po dun sa surface nung ating nanoparticles wherein yung proteins naman nung ating pathogens ang mag-interact. So para siyang lectin, yung protein carbohydrate interaction. That's what I'm thinking, and that's what the collaborator mentioned to us. The chemistry behind yung surface nung pathogen. In general, yung mga pathogens daw, most of them have similar surface chemistries. And now when we proceed to the DNA-based detection, that's when we can distinguish among the pathogens. So I hope I answered yung question po ni Dr. Humawan.
Yes, ma'am. That question po from Dr. Larry din po, our PSDB president, Doc. Another question, ma'am, from Ma'am Vay Casison, what's the advantage of nanobiosensor technology over the usual dipstick ELISA kits with regards to field application? Have the two methods been compared?
Okay. Yeah, thank you for the question. For the ELISA technique, antibody-based usually po siya. Just like in COVID detection, we have RAT and the PCR-based. So for ELISA, well, mas mataas po na magkaroon tayo ng false positives, false negative na mga results. Well, for the nanobiosensor that we're using, specifically the one that we're developing, it's DNA-based. There are some laboratories as well who works on antibody-based detection. Still nanobiosensor, but their receptor is antibody. So yun po yung kaibahan ng aming technique. It's DNA-based, and for ELISA, it's antibody-based. Ayun po.
Thank you,
So
ma'am.
we still have to validate. That's the next stage of our research. We compare with ELISA and the PCR-based technique, the standard methods, along with the plate method or culture method.
Yes, Doc. Noted po yan. Eto ma'am, magandang question to ma'am. From Dr. Nadia again. Is the use of nanobiosensors for the detection of dengue in mosquitoes already being used by DOH or RHUS, or does it still need to be commercialized?
Okay. About the dengue nanobiosensor, we just completed the phase 1, which is the proof of concept. So it was funded by DOST PCHRD. The plan is to pursue yung validation part po. So we're still in the R&D part. But I'm glad na dun po sa proof of concept namin, we were able to detect yung actual samples from St. Luke's. For clinical samples from St. Luke's. So there's ano ba tawag dito? There's a potential talaga for this nanobiosensor. So hopefully makalabas na nga siya.
Another question, Dr. Fernando, from Iken Nico P. Labnau, Silliman University. Here's the question, ma'am. Estimatedly, how fast will be the whole process of each of the two detections of the proposed nanobiosensor kit?
Okay. So in terms of, well, I mentioned earlier that for the actual detection, the electrochemical detection, less than 1 minute, the plan is for us to have the DNA extraction done in less than 1 hour. In less than 1 hour on site. It's actually a challenge. So, ayun po. So, mga roughly 30 minutes to 1 hour for the detection, along with the sample preparation. Sample preparation and detection on site.
Okay. Thank you po. Okay, another question po from Miss Margaret de Guzman, UP Manila. How do you dispose the nanobiosensors after their use? Because of their nano size, they might find their way into the aquatic or terrestrial ecosystems.
Okay, yeah, that's a very good question. We have a waste bottle for our, I think she's pertaining with the nanoparticles. We have a waste bottle for each nanoparticle, and we're also working on the regeneration of these nanoparticles para magamit namin for the sa mga iba-iba pang devices, So for now, we're not disposing it. Hindi namin siya binibigay din do sa mga kino-contract ng university na nagdi-dispose ng ating mga laboratory waste. It's still in the laboratory, And as I mentioned, the plan is to regenerate the used metallic nanoparticles. And yeah, we're concerned with the safety as well, nung ating, with the safety, nung use natin nung nanomaterials.
Okay, ma'am. Then follow-up question po from Dr. Larry again. What kind of pathogens or substances can be detected using nanosensors? Is it viral, bacterial, fungal, parasites, and also specific chemicals po ba can be detected?
Okay. As far as our team is concerned, we focus on bacterial, viral, fungal pathogens, and there's another team which I'm also a part. Sa ano po sila, sa animal science, they detect salbutamol in feeds. So salbutamol is a compound na usually po nagagamit daw nila para lumiit o kumonte yung fats ng ating mga binibiling mga meats. So instead of DNA-based detection for that application sa mga chemical contaminants, they make use of antibody-based detection or other platforms, hindi po DNA-based. Ayun po. So thank you po for the questions.
Okay, then last, ma'am, siguro last question na to. We have senior high school students, ma'am, or STEM that are watching po live in the PSDB YouTube. Though part of the DepEd senior high school curriculum is the research subject. My questions are, ma'am, what can you advise or tip, sabihin natin motivational inspiration to our future scientists and researcher, itong mga Gen Z generation po that are conducting and studying scientific research po?
Okay. Sa mga students natin, sa mga high school students, nung ka-age ko kayo, siguro malaking factor yung motivation ng mga mentors nyo and even your family. But think of something that nag-start kasi yun sa motivation mo. Think of something that you can contribute. Minsan yung iniisip natin ano bang purpose ko? Ano ba yung pwede kong ma-contribute? And minsan minsan magugulat ka, hindi ka pala scientist, artist ka. So iba-iba. For those who are tingin nila, nafe-feel nila into science sila, then ayan, ano na yon, automatic tingin ko, na biglang ting. Ano ba yung mga pwede kong gawin? Yung iba gustong maggawa ng mga devices at home. Yung iba, they want to, lalo na mga generations ngayon, they watch videos, And from that, pwedeng matickle yung mind nyo on how, on what you can do, pwedeng research siya or something na yung iba nga entrepreneurial na. I do encourage you, to do something na pwede niyong pagkakitaan. You can do research at home siguro, with your, kung ano yung available at home. Lalo na ngayon na pandemic pa, na pwede niyong pagkakitaan. So again, it starts from your heart. Kung ano yung gusto niyong ma-contribute and think of something on how you can work on that. Ayun po. Thank you.
So, pwede tayo mag-hi, ma'am, sa YouTube. Nandiyan po
Hi,
mga
hello
students.
po sa lahat. I hope you learned something new from what I've shared. Thank you po. Hello po sa lahat.
Then sa comments po natin, marami po tayong mga participants na nag-thank you din. Ang dami din po nilang natutunan, ma'am. Then proceed. Last question na po yun. Then proceed na po tayo for the awarding of certificates, ma'am. Okay, so the Philippine Society for Developmental Biology Incorporation, Co-Institute of Biology, College of Science, University of the Philippines, Diliman National Science Complex, Diliman, Quezon City. This certificate of appreciation is given to Dr. Lilia M. Fernando for the generous sharing of knowledge and expertise as resource speaker during the PSDB 2022 Webinar Series Session 1, held online on April 23, 2022. Signed, by John Vincent G. Navalan, Program Chair, Dr. Dino T. Tordesillas, our Secretary, and last, Dr. Larry Ann Ipulan Colet, our PSDB President. Thank you, Dr. Lilia and Fernando.
Okay, thank you po, and congratulations to PSDB.
It's your turn now, Dr. Dino.
Okay, maraming salamat po, Sir Jodel, for that, for leading our open forum number 1. So, ano po, salamat din po, Ma'am Lilia. So, ang dami po naming natutuhan ngayong hapon. And also, for that very inspiring talk for our future scientists that may be interested to go into STEM or to STEAM. Di ba kasama yung agriculture dun sa, ano natin, dun sa field natin na yun. So ngayon po, we have this chance for our first photo session for this afternoon. Babalik po si Sir Jodel mamaya sa ating open forum number 2. So ngayong we will have our first photo session with Ma'am Lilia because she's very busy. Kailangan po niya umalis agad. So mag-photo op na po tayo together with Ma'am Lilia. So with to our around 140 guests this afternoon. So please turn on your cameras if you are able to, and we will take our photos. Sir Dano, will you be taking the photos for Okay, sige po. Hintayin po natin magbukas ang mga camera ng ating mga participants ngayong hapon. We do have a lot, around 140, kung hindi po ako nagkakamali. 140 plus. Okay po, yung mga humahabol, habol na.
Alright, sige po. Magpipicture na po tayo in 10 seconds. Paki-on po yung mga camera and also in-upload po natin yung virtual background for this webinar series. So kung wala pa po kayo, pwede niyo pong i-download.
Sige po, habol
Okay.
po yung mga on na ng camera.
Yan. Mahabol. Nagbubukas pa po ng camera yung karamihan. So hintay-hintayin natin ng konti. Okay. So, okay. I think we can start. So we have 4 panels po. So i-hold nyo lang ng matagal-tagal yung smile ninyo. Ayan, shinare po ulit ni Sir Jashin yung virtual background natin. So pwede nyo po siyang i-download. And you can use it para sa photo ops natin. So meron ulit po tayong photo ops later after the second speaker. Alright. Okay, pwede na tayo mag-start. So again, we have 4 panels. So pahintay na lang po. Paki-hold ng medyo matagal-tagal yung smile. Okay. Okay, we can start. 3, 2, 1. Smile. Another one. Okay. Next panel. 1, 2, 3. Smile. Another one. Third panel. Smile. 3, 2, 1. And then we have our last panel. 3, 2, 1. Smile. Okay. Okay na po, Sir Dino.
Okay, maraming salamat po sa photo op at syempre sa ating speaker of our first speaker for session for our first speaker for this session, Dr. Lilia Fernando. Maraming salamat po. Let's give Dr. Fernando a virtual round of applause. So maraming salamat po. Okay, salamat po ma'am. Salamat po ulit.
Okay, thank you.
Okay. Bago po tayo, before we proceed to our next speaker, let me share the presentation again. So, again, this is that was just the first of our two speakers. Dr. Fernando was just the first of our 2 speakers this afternoon. Again, this is welcome to the 140 plus 140 something attendees here in our Zoom session. And to those who are watching us on YouTube, again, thank you for supporting our PSDB 2022 webinar series, and thank you for tuning in and being a part of this activity of the Philippine Society for Developmental Biology. Again, the PSDB 2022 webinar series is presented in cooperation with MaCare and sponsored by Merck, MedTest, and PhilAnaserve Incorporated. Okay. Now, from Cacaos, let's go to Carabaos. Okay. Our next speaker is
Sorry,
Dr.
excuse me, Sir
Euphrosine.
Dino. Okay.
Yes. Yes,
May
ma'am. Go ahead.
ano po doon sa, may mga block parts po doon sa share na screen.
Oh, sorry.
Okay po. Thank you.
Yeah, I think yun. Okay, so, sige. Okay, so, ayan, mas okay na po ba?
Sa upper part na lang po, pero we can see Dr. Atabay's info na po.
Okay. This is, I think this is something to do with my internet connection, so please bear with me. So again, for our second speaker, we have Dr. Euphrosine P. Atabay, and she will be talking about enhancing livestock production through reproduction research and innovations. Dr. Atabay is a technical expert on animal reproductive biotechnology and physiology and is actively engaged in research focused on enhancing female fertility and reproduction in water buffaloes with application in other livestock production. Her expertise include buffalo and cattle semen, oocyte and embryo cryopreservation, in vitro fertilization, timed artificial insemination, and embryo transfer. She's also engaged in research in molecular aspects of animal fertility and reproduction. She's a key partner in the development and aggressive implementation of the intensified reproductive management program in livestock, which primarily involved the use of modern biotechnology to maximize animal productivity. She underscores enriching understanding on buffalo reproduction to achieve more precise timing of ovulation and insemination to improve pregnancy. Apart from her immense involvement in science and technology activities, Dr. Atabay also serves as the focal person of the Intellectual Property Management Program of the Department of Agriculture, Philippine Carabao Center. She is hugely instrumental in the establishment of the IP Technology Business Management Office of the agency and of a system which facilitates translation of research into products and services for the benefit of the clients, livestock industry, and of the general public. So without further ado, we have Dr. Euphrosine P. Atabay talking about enhancing livestock production through reproduction, research, and innovations.
Good afternoon, everyone. So are you hearing me?
Yes, ma'am, we do hear you loud and clear.
Anyway, so can I be allowed to share my screen? Okay. Okay, so magandang hapon po sa ating lahat, and thank you, Dr. Tordesillas, for that very generous introduction. So again, I wish to greet all the officers and members of the Philippine Society of Developmental Biology. So again, I wish to welcome you all to this session 1 of the 2022 webinar series of PSDB. So I am being Atabay of the Philippine Carabao Center, the Department of Agriculture, and today I'm so much honored to be able to share with you all, especially our attendees, in this topic, or our work in enhancing the livestock production through reproduction research and innovations, and with focus on buffalo reproduction. So we expect that at the end of this presentation, you will be able to recognize the importance of water buffalo in the Philippines, and identify some problems that are associated with buffalo reproduction, and then what are the reason, or identified some factors that lead to its transformation from just a draft animal to a dairy animal. And of course, be able for us to understand, especially our students, what are the programs, projects that are at hand in order to develop the dairy or the livestock industry. So, do you know that there are two types of water buffaloes in the Philippines? So we have the swamp or dairy type, and the riverine or dairy type. And as to the chromosome number, the swamp has 48, and the riverine has 50. So the crossbred, which is around 49, is considered fertile. So the scientific name for buffalo is Bobalus bubalis, and they are both called the Philippine water buffalo. So in how do you say that? In locally, in Ilocano, they are called nuwang. In Kapampangan, they are called damulag, and maybe in Visayas, they are called carbaos. But anyway, this is a very interesting animal for us, and that is the very essence of my presentation today. So for me, the water buffalo or the carabao is not just a draft or traditional animals, because through time it has improved. And so initially, this carabao has been part of the farming or agriculture, especially with our farmers. So these are even called our national animal, and they symbolizes resilience, hard work, and efficiency. And that is why they are called the farmer's best friend. And they are partners or they're faithful companions in farm work, such as plowing the field, or tilling the soil, plowing the field, and even transporting farm products. So these are really the reasons why carabaos or water buffaloes are simply called as the beast of burden. But their transformation, of course, initially from just being the beast of burden to a beautiful beast of burden has been demonstrated. So, and they are very much part of our culture and festivities. So this is initially the cultural aspect of the importance of carabao. So in particular, we have, they are part of the kneeling carabao is part of the festival in Pulilan, Bulacan. So wherein they are groomed and then decorated and then paraded during the town fiesta, and then before the church, they are trained to kneel in order to honor and give thanksgiving to the patron saint. So this is the kneeling carabao. And we also have the Nuwang Festival in San Agustin, Isabela, and wherein the Carabao honors. So these are our partners in our Carabao development program, no? So usually during Nuwang is how they call Carabao in Ilocano. So during the Nuwang Festival, they parade their animals, and they are happy and proud to as part of their achievement and active participation in the Carabao Development Program of the Philippine Carabao Center. So, and then of course in Visayas region, we have the Carabao Carosa Festival, wherein, this is specifically in Pavia, Iloilo, every third day of May. So there is this carosa and different, well decorated. And then another activity is the beauty queen, or the parade of some beauty queen, and normally it ends up with a Carabao race. So this has become a long time and annually celebrated festivity in Iloilo. Yes, and of course, Carabao painting is one of the pride of Ilocanja, no? So, and here they are part of their culture, for their painting the buffaloes, first the white, and then come up with a living canvas. And then maybe, recently in, I think this is in Negros. So part of their important town celebration, they are also painting the Carabao to reflect their tradition and culture. So all of my, the things that I mentioned are reflective of the buffalo as initially still an important part of the cultural aspect. And now, so what else can buffalo has to offer? So aside from those I mentioned, through time, the buffalo or Carabao has continued to become interesting, being an important source of milk and meat that provides especially the livelihood and source of income to our farmers. So essentially their transformation from being a traditional draft animal into socio or economically valuable animal is the important transformation or happening in the Department of Agriculture, or particularly in the dairy or livestock industry. So, okay, just to give you some backgrounder on what is the current development in the livestock or dairy industry. So buffalo has the current herd inventory is around 2.9 million. And with regards to how many is pure or dairy type, it is around 80%, and 20% only for the draft or our native one. So that's why through the years, it is able to increase or to contribute a larger part, or 35% of the total local production. So it's getting higher and higher through the years. Well, of course, the cattle being able to produce naturally high, this is the major contributor. But the buffalo with our program is getting or increasingly contributing. Now, in terms of the milk quality from the buffalo, so take note that its nutrient composition in terms of protein, fats, carbohydrate, energy, calcium, the Carabao's milk are higher in percentage compared with the cow's milk. So for example, the protein is 4.3 for buffaloes, whereas in cow it's only 3.2, and so on. And also comparatively with goats, buffalo has higher percentages of this nutrient composition. So that is why buffalo's milk commands a higher or prime price compared with other dairy species, no? So, but then currently the reproduction and milk production in buffaloes is faced with a lot of challenges. And this is due to some management program or management practices, but with specifically, of course, in the reproduction challenges. So how does the Philippine Carabao Center address those problems in buffalo reproduction? So before I give you the answer, allow me first to give you a glimpse of our the workplace, the Philippine Carabao Center. So we are here in Nueva Ecija right now, and it's where its headquarters is present. So I hope you had been here. And then it is an attached agency of the Department of Agriculture, and we were created by the Republic Act 7307, otherwise known as the Philippine Carabao Act of 1992. And then actually 10 years before that, we were just a project coordinated by PICARD, but funded under the FAO, UNDP. So now we were institutionalized to Philippine Carabao Center, and also because of our accomplishments, we were given additional mandate by the Department of Agriculture and with A08 of 2009 to lead the livestock biotech program. So, and also we take pride in our being a few of the agencies that is ISO certified, so for the integrated management system. So what is our mission? Of course, we must be able to conserve, propagate, and promote the water buffalo as important source of milk and meat towards better nutrition and to improve the general well-being of the rural farming communities. So through our milk, and the meat, we must be able to improve or help our farmers. So in order to achieve the mission, the PCC has developed this, what we call the Carabao Development Program, which is our banner program. And this is implemented by 12 regional centers all over the Philippines. So in here. And of course, we have partnership with universities and local government and regional offices. Now, the Carabao Development Program has 3 major components. So we have the Genetic Improvement Program, wherein we talk about the riverine buffalo or the dairy buffaloes, the swamp buffalo. We talk about also the conservation, and then the upgrading through some important strategies, artificial insemination like that. And then of course, this is the animal aspect of the program, and then we have this the socio-economic aspect component of the CDP. So we have the buffalo-based enterprise. So in here we develop cooperatives and federation, and we give them access to credit and provide them market assistance. So milk is just Carabao is just producing a small volume of milk. So, but when we do value adding through this work, and then we are getting more, our farmers are able to gain more incomes. And of course, the very important component of our program is the research and development. So we develop technologies and protect our technologies and be able to also provide, commercialize them. And recently we are into that direction, commercialization and utilization of our technologies. So since what is the role of reproductive biotechnologies, which is really the heart of my presentation. So of all the components, we are involved in the genetic improvement program. So to be able to reproduce the buffaloes, to be able to improve their breeds, we are using the reproductive biotechnologies. And of course, we are anchored on a well-defined breeding program. So, breeding program involves a very effective and efficient selection of the breed of animals. And we work together to be able to multiply using the reproductive biotechnologies. So to answer the great demand for animal as well as production. So I think that's the important technologies in here. So also, and specifically when you talk of the reproduction program, we are also developing some, recently we developed this intensified reproductive management program. So we call this the Triple-E strategy. And what is the Triple-E? Means we have to enhance the pregnancy rate for more animals through various techniques, the artificial insemination, ovum pickup, and IVF. And then another E is after insemination, we should be able to conduct early pregnancy. So we will not keep them long if they are not pregnant. So we use these techniques, ultrasound and other tests. And then of course, the effective rebreeding program. So when you find out that the animal is not pregnant, so put it back to the breeding herd. And essentially we are anchored in the understanding the ovarian physiology. So para mas ano natin yung proper timing. Alam mo sa reproduction kaya hindi kuminsan nagkakaanak, kulang ng timing. So for us to address that, we should be able to understand the very aspect, the physiology under to be able for us to understand the underlying mechanism and physiological events. Ayan po. So now I just to give you the highlights of some technologies. So as an animal reproduction, ito po yung the earlier we use the artificial insemination, yung di likas na palahi. So we are using the cryopreserved semen here. So and then in here, this is the fetus, and then detected through the ultrasound. So ito po yung, when we talk of reproductive biotechnology, we were trying to categorize this into a female or male. So this one, using the semen to spread or disseminate the parental genetics, artificial insemination. So, but then, what are the challenges? Ito yung very, buffalo is more difficult, more challenging to reproduce compared with cattle. So ito yung nila, the poor, what are the problems dealing with buffaloes? Yung poor estrus manifestation, yung parang kahit na sila ay estrus na or nagsusyo na ready na sila, hindi makita. So they are prone to the so-called silent heat. Yung ganun, kahit na bantayan ng mga laborers namin, and normally they are showing heat in the night, and nakakatulugan. So it's that major problem. And of course, yung ovulation or release ng egg from the follicles ay hindi may timing sa similya, So isang problema pa yan. And seasonal infertility, masyadong nai-stress sa heat yung ating animal during summer. And then pag nanganak na sila, ang hirap na ulit na mabuntis, maybe because of some negative energy balance. And medyo delayed sila bago mabuntis. So this will essentially result to low conception rate, low calving interval, and low production efficiency. So yun yung mga obvious na dahilan kung bakit may problema tayo or mabagal, no? Kaya kami pinapagalitan ng ni Lola. Mahina, mabagal daw kami sa dairy. So that's precisely more difficult. At matipid pang magbigay ng gatas ng buffalo. But then, aside from all this, allow me to emphasize also some intrinsic factors or yung inherent sa buffalo, no? So, have you seen already the ovaries? Nakakita na ba kayo? Or maybe this may be your first time to see the ovaries. This is the ovaries ng cattle. So, as I was saying, no, mukhang mas sila, masigla. They look bigger, and then ito yung mga follicles na sinasabi ko na may egg yan na i-release. So if you look at that, napakadami, maraming ang cattle. And when you open up, ito yung mga structures nila, have you seen the corpus luteum? Ito yung nagme-maintain ng pregnancy. Ito yung nagpo-produce ng hormone na progesterone para ma-maintain ng pregnancy. But comparatively, look at the buffalo, halos gray and parang no structure, napaka-dull, and yung color niya is really pale and small. So ganyan ang itsura ng buffalo. And in terms of ano ba yung mga structures na makikita natin sa ovary? So ganyan din po yung mga ovary ng mga tao. So ito yung sinasabi natin na mga follicles sa at birth. Yung buffalo ay mayroong 12,000 lamang, samantalang ang cattle, yung pag ipinanganak mo na yung guya, no? Ang cattle ay more than 50,000. And then yes, in terms of ovary size, small talaga ang ovary, malaki talaga ang cattle. And yeah, so and the small number of follicles in buffalo is being aggravated, parang pinapapalala pa kasi monthly nagkakaroon tayo ng yung buffalo, may sinasabi tinatawag tayong estrous cycle na kung saan 21 days siya. So nagre-release siya ng egg. So that's practically a stage or in the process, no, from the very start, from the birth, no, nagkakaroon na siya ng follicular atresia. So it's essentially the death of the cell or the follicles. So just a review, meron tayong mga initially we have a lot of thousands or primordial follicles, primary follicles, secondary follicles, hanggang siya ay maging ovulatory follicles or sinasabi natin ito yung final ovulation. So in the process, talagang ang daming napupunta. So we call it the follicular vestige. And yun na nga, and this is the one that aggravates the problem doon sa buffalo. And sinasabi nga dito, the buffalo have a follicular atresia of 67%, whereas only it's 50% in buffalo. So that's why dito pumapasok yung importance or the dramatic effect of the technology kasi parang bago yan mag-atresia ay may magagawa ko na para mapakinabangan sila. So yes, that's the technology are helpful, valuable, and magical talaga siya. Just to give you some the basic in reproduction, kahit na naman sa human, no, ganito talaga. So when you talk of, di ba na-mention ko kanina na in every estrous cycle at the end may ovulation. So ito bale na ang kalabaw ay may 21 days estrous cycle. So tapos yung phase ng cycle, may tinatawag tayong luteal phase and follicular phase. So pag luteal, and then mayroon tayong tinatawag din na sa bawat cycle pala, may mga wave, follicular wave. O katulad nito, we have an animal or a buffalo that has 3 wave cycle. So 1, 2, and 3. So itong first two, ito yung mga stages ng follicular development. May recruitment, may selection, may dominance. Pero nagre-regress lang siya. And then another, recruitment, selection, dominance, and then regression. But on the last, ay yun yung tinatawag nating may ovulation na. So we call that the follicular phase. So kung just to emphasize, ano ba yung follicular wastage? Ito lahat natatapon, walang nangyayari. So sa 3-wave cycle, lahat ng follicles dyan, tapon-tapon. And only one at the end of the cycle is ovulating. Pag hindi pa yun na timingan, sayang. So ilang beses nagsa-cycle ang kalabaw. And every time there's just 100 of so ganun din sa tao ang So this is just a follicular wastage, just to emphasize na may magagawa ang reproduction. So may mga animals naman na 2-wave cycle lang. Yan, ayan, recruitment and then regression and then recruitment and successful ovulation. Just to emphasize. So ayun, so ano ba yung mga ginagamit namin mga technology, no? So we do the estrosynchronization and artificial insemination. Yun nga yung sinasabi namin na to maximize the use of the semen or the frozen semen. Sarang sinasabay-sabay namin na ma-induce yung estrus or inhit ng buffalo. So we do the injection day 0 and then okay, sorry for the term, mag-i-inhit na siya o maglalandi na siya after 3 days. And then we do AI, 2 times AI. So that is the very basic artificial insemination. We do synchronize the females. So in here, you see may dapat mag-develop siya ng mga signs of heat, like the mucus discharge, the color of the vulvar opening, something like that. So this is artificial insemination. And the associated technology for us to be able to perform or bring the sperm samples to other different parts of the country, we do the semen cryopreservation. So it's a process wherein the buffalo sperm are stored in liquid nitrogen. And nowadays, we are evaluating using the computer-assisted sperm analysis. So this is more objective, more accurate, ensuring the quality of our produced frozen buffalo semen. Yeah, so that's for the male, and then this is also optimizing the female good genetically superior buffalo. So we call this the ultrasound guided ovum pickup. So normally kumukuha lang kami sa slaughterhouse ng mga oocyte or sa ovary, but in here talagang identified na superior yung female animal. So we do this by ovum pickup. So may machine siya na i-deposit and then withdraw or aspirate the oocyte from the follicles, and then we do the in vitro fertilization. So ito na po yung mga sample ng mga oocyte cumulus cell complex aspirated from the live animals. And these are the embryos formed. And then this embryo can be cryopreserved and also transferred. So that is, we call this the ovum pickup. And this is also done in human, no? To address infertility. So one of our recent technology is, we call this the fixed time AI, or yes. So recently, gumagamit lang kami ng prostaglandin and then do AI na. Ngayon, ito yung talagang naging problema sa buffalo. Parang hindi matimingan yung ovulation kung kailan inire-release. So that's why with Kanina, makita nyo na ang sinasabi ko ay yung estrus synchronization. Ngayon, ang sinasabi ko is ovulation synchronization. So, parang ini-induce talaga, hindi lang yung estrus, kundi yung ovulation. That's why, so ang result talaga nitong ating tinatawag na fixed time AI, napataas niya yung conception rate, and then yung ovulation rate, napapatama niya. And basically, this has been applied all over the country. So we initiated this technology in 2014 at PCC, and it's now more than around 8 years already. But the most important associated technology when you talk of the fixed time or timed insemination is the use of the transrectal sonography. So napaka-importante yon kasi you know, in agriculture, we should be dealing with things in precision. Ano ba yung throughout the treatment or hormonal treatment and preparation, we should be able to evaluate or monitor the growth of the follicle. So ito dito, for example, in here, 10.6 millimeter, and then 13.8, and so on. So that is very important for us because this will dictate that it's now high time for us to do the insemination, because with the size, the ovulation is expected. So normally, mag-AI ka muna, paunahin mo muna yung similya, mag-travel sa and then saka pa lang mag-release. And yes, true enough, the ultrasound also is able to allowed us to do the early pregnancy diagnosis. So ano yung kaiba? It's also important na when you talk of intensified reproduction, dapat pag nag-breed ka or nag-mate ka, dapat ma-detect mo kaagad kung buntis or hindi. Kasi kung hindi, dapat mabuntis mo ulit. Ibalik mo na siya sa breeding herd. So this is just to emphasize na sa aming pag-aaral on the ovarian physiology, because napaka-importante yun eh. So we focus on the size talaga sa bawat protocol na day 0, day 7, day 9, day 10, ito na yung pag-AI. So ito, mag-inject ka ng hormone sa day 0, another on day 7, 8, and 9. But ano ba yung ina-expect nating follicle size dyan? So these are the critical points. So pag ovulatory na siya, malapit ng ina-expect na natin na 12 to 13 millimeters. So the follicle has already achieved the right size, and prepare na tayo for insemination. So I mentioned that our gold standard for detection of pregnancy is using the ultrasound, but recently we are also using this kit, pregnancy kit. So the pregnancy associated glycoprotein kit. So na-mention nga kanina ni Doc Lilia that this is ELISA-based or antibody-based technology. So para na rin yung colorimetric, no? So this is a commercially available one, but you see the ultrasound can detect pregnancy at around 35 to 40 days, but this PAG kit can detect as early as 25 to 30 days. So parang pag empty na yung animal, abangan mo na sa susunod kasi 21-day cycle. Agad, buntis agad, no? Something like that. So that's the beauty of the early pregnancy detection and all these technologies. And of course, right now, we are also into developing the nano-based kit, no? So that's why I'm also interested with the presentation of Dr. Fernando, because essentially it's the same direction. The development of nano-based technology for pregnancy. So Ayan, so ito na po yung mga products namin for the fixed time AI. So, you know, when you do synchronized or sabay-sabay na pag inseminate, you're able to produce this kind of sabay-sabay din na age group. And then at some point, we are able to produce twins. And then this is our crossbred, no? Yung sa katulad sa Nuwang, doon sa Isabela. So it's just 50-50 of the native and dairy. And then we also use the technology in cattle. So we are also into the livestock industry. So whatever we produce, the technology that we use in buffalo, we use that in other species. So may tinatawag din tayo na multiple ovulation and embryo transfer. So ito kung doon sa normal insemination natin, we only expect one oocyte or one ovulation. Dito naman po, we are expecting a lot or multiple ovulation, hindi lang single ovulation. Because, and then when you talk of embryo transfer, you talk of the donor animals, wherein that is hormonally injected or prepared. And then we do also the AI, and then we collect in here, we collect the embryos, and then we evaluate the embryos, and here are the samples of the embryos that we produce when collected, and then some are frozen and some are transferred. So also there should be the recipient animal or the surrogate animal to receive those embryos. So ito lang naman yung difference, no? Ito yung ovary pag maraming follicles or corpus luteum, no? Alam mo, sa una, follicles muna. Pag pumutok na yan, yung pinanggalingan ng follicles nagiging corpus luteum. Yung corpus luteum nagpo-produce ng progesterone para ma-maintain ang pregnancy. So ito sa multiple, marami tayong injections ng follicle stimulating hormones para mapadae, ma-maintain yung maraming, you know, yung the wave of follicle. So, and with the fixed time MOET, multiple ovulation, we were able to produce pag-asa. Yes, I think this is very significant, and you know, when we transferred one embryo, pag-asa is the very first calf born out of the multiple ovulation and embryo transfer. And you know, it's very historic or very memorable because tomorrow will be his second year. So Pag-asa is a male one, and he is now being trained to become the semen donor. So galing po siya sa multiple ovulation at may mga kasama pa siya for transfer. So yan, naalala ko, birthday bukas ni Pag-asa. And we also do the fetal sexing, no? Isa pang kagandahan ng ultrasound ay makikita natin through around, na-optimize na namin to sa buffalo at around 60 days post breeding, ay makikita na natin kung siya ba ay a girl or a boy. So through the different anatomical landmarks. Ayan. So it's a girl, it's a boy, something like that. No? May implication din yan. So that's fetal sexing technology. And also the recent one is, ito na po yung pinaka-interesting one, no? When you talk of the dairy, the very challenge of increasing our dairy or improving our yung milk production locally. So nowadays, the use of sex semen, ibig sabihin yung X chromosome bearing sperm ay napoproduce na po yan, or we call that simply the sex semen. And of course, when you use that, you are producing predominantly female ones for dairy. So ito yung semen sexing technology, and this is patented one. So ito yung pinaka-basic principle niya. So you have to stain the sperm, and individually it has to pass through the the spectrophotometer, and because of their whether they are X or Y, they are being separated. So yung depende po to sa intensity ng illumination. So because we are providing what call that, we are staining the DNA. So sabi nila pag ang babae nag-absorb siya ng maraming stain, and then when it pass through the laser, it will illuminate far more compared with the male. So by virtue of the intensity of illumination, na nagsiseparate sila. So I think this is ito na talaga yung pinaka-ultimate ano natin na when we talk of dairy. So now what are the developments? So Department of Agriculture or being led by PCC for us to be able to acquire or to have this patented technology operationalized here. Napakamahal po nito, and I think that the US, we are now negotiating with the cement technology company in the US for us to be able. So sana ipagdasal natin na mabalitaan nyo na lang next year or in the near na mayroon na tayong sex cement sa buffalo as well as in cattle para mas mabilis po ang ating pagdami ng mga gatas. Well, of course, sa baka ay nabibili na lang po ito through the National Dairy Authority. So sila naman talaga yung mandated for dairy, no? So we asked for some samples of the sex sorted semen, and true enough, si Liwanag was born last March 2021, just last year. So talagang babae siya. So this is the Liwanag, the hope, no? That one day this will happen in the Philippines. So how are those all our programs in reproduction at PCC? How are we aligned to the national level program of the Department of Agriculture? So I must say that we are one with one DA through these strategies. No, so we one DA agenda, and we have the key strategies towards transforming the Philippine agriculture. So ito yung major components, no? Consolidation, modernization, industrialization, and professionalization, and the use of the reproductive biotechnologies as well as other technologies at Carabao Center is under modernization. So in here it says the technology and innovations as one of the strategies for one DA. So we are so much aligned into that national program of the Department of Agriculture. And also recently we developed this Carabao Industry Roadmap for 2022 to 2026, or even the dairy industry roadmap. So the use of reproductive biotechnologies, both for females and especially the use of sex semen, the use of multiple ovulation is very much a part or evident in these roadmaps. So this major, this is basically a medium-term plan we developed, and this also covers some policies, some regulations in order to sustain, strengthen, and drive the economic growth of the dairy or livestock industry. So, so in closing, I would like to emphasize that the use of all this reproductive biotechnology can accelerate the production of animals that can produce milk, meat, as well as to increase the productivity and the profitability from the Carabao-based enterprise. And this ultimately will lead to our empowerment and prosperity of the farming communities. So with that, thank you so much, and I hope I'm able to share what's happening in our department, especially at PCC, with regards to our effort on research as well as innovations on biotechnologies and reproduction. So thank you so much for this privilege to be part of this prestigious society. Thank you.
Thank you very much for that wonderful talk, Dr. Atabay. I was able to visit the PCC way, way, way back when in the 90s. And we were told that the PCC was at the forefront of research way back then. And it's nice to know that it is still at the forefront in the research and innovation when pertaining to not just Carabao reproduction, but
Yes.
everything related to Carabao sciences.
Yes, so many fields still, and we have advanced, of course. So we would like to share further later.
Okay po. Salamat po, ma'am. Again, now we have, we welcome back Sir Jodel for our open forum number 2. Sir Jodel, take it away.
Hello, Dr. Atabay. Rinig, ma'am?
Yes, sir.
Okay, magandang tanghali po, ma'am, at saka sa mga participants natin na still nakikinig at ang daming learnings pong natutunan.
Congratulations, blockbuster yung attendance natin.
Okay. Yes, ma'am. For the Q&A po, Dr. Atabay, ito po yung first question natin. From Noreen and Factor. Are there significant differences in terms of reproduction between Carabao and Tamaraw? If yes, what are these specific differences?
Yeah, of course, partly or genetically, I think they belong closer to the same genus, something like they are within the same family. But of course, in terms of the reproduction, basically the same. But we also tried. We were engaged also in the reproduction in Tamaraw, but because of their nature, wild nature, and they're in the field, or they were bred at one point in captivity, but it was not successful because we cannot apply any reproductive biotechnology. You cannot collect the semen. So before you can approach to tranquilize them, to collect semen, maybe it's really a difficult thing to do. So I believe that whatever technology that works in buffalo can also be applied in Tamaraw. It's just that they are just dangerous, they are just vicious, and I think it's better to let them reproduce in the wild rather than to put them or hold them in captivity. So basically, our experience was a failure.
Thank you, Doc. Second question, Doc. Medyo nahiya po di po na lagay yung names ni Ma'am or Sir. It's the Department of Agriculture or Philippine Carabao Center promoting Carabao breeding in all provinces. Which provinces in the Central Visayas have the capacity for Carabao breeding?
Oh, yeah, as I mentioned, our regional centers are the one implementing this. So all over the Philippines. So of course, we have 3 centers or 4 centers in Visayas. The WVSU Visayas and La Carlota Stock Farm and Ubay Stock Farm, something like that. So we have all centers there doing artificial inseminations, doing the other business or Carabao-based enterprise program. So sa mga university, mayroon din po sa West Visayas State University and La Carlota sa regional offices. So mayroon po tayo sa Visayas.
Thank you, Doc. Another question po. How long does it take for a female carabao to become fertile again after giving birth to a calf? At what age does a female carabao become reproductively mature?
So yung tinutukoy natin yung puberty age niya, pubertal age. So
Yes,
actually, yun na yung normally dapat kasi 2 to 3 years old ay maging productive na or breedable na yung ating buffalo. But then, siguro may mga factors minsan sa nutrition, medyo nadidelay yung kanilang breeding, no? Yung ability to get impregnated. So that our aim really is to at least 2 to 3 or buntis na sila, and also depending on their weight. And then also yung reproductive status nila, no, na dapat yung cervix malaki, naman na, kasi without that ay kawawa naman. So they should be breedable by age, by 3-year-old. And then the second question was at what point can they be rebreed after giving birth? So actually after 60 days, sila pinanganak, and then hayaan mo muna after 21 days, i-allow mo munang and then the second cycle. So pwede mo na siya mga 40 days after birth, by 60 days, pag pinalpate mo siya, pwede siyang buntis ulit. Kung na-AI mo siya ng 40 days postpartum, ang tawag natin. But then, again, may mga, because of some, sinasabi nating negative energy balance, nadidelay yung kanilang postpartum estrus or yung ovulation. So, but because of our intensified reproductive management program, we have the 60-day breeding program. So dapat pag in-ultrasound mo siya sa 60 days, buntis na ulit. So of course, we do this hormonal induction, we do the good feeding and health management. Kasama yan.
Thank you, Doc. Question pa po. Can undergraduate students do on-the-job training or research apprenticeship at the Philippine Carabao Center?
Yes, normally we do that because as I said, we have also centers that are university-based. So not only here at here in Nueva Ecija, but we have a UPLB and wherever. PCC centers, which are university-based, so they can just go there and approach for possibility of researchers. Yes, because we are all encouraged, and that's part of our operation to do research.
Thank you, Doc.
But of course, it was specifically here in the center or in the central office. We were hampered by pandemic. But we're starting.
Yes, Doc. Question po from the participant again. How does the Philippines Carabao Center minimize the stress experienced by female carabao during artificial insemination?
Yeah, stress by artificial insemination. Normally naman, we are providing the appropriate restraining tooth, something like just to keep them behave, something like that. And then the animals are already used to do these things. Now, if we do this in the field, we advise the farmers to provide the chute. na lalagay doon during artificial insemination to avoid the animal jumping and then to avoid being hurt during AI. So proper restraint. That's part of our requirement.
Yes, Doc. Follow up to Doc, do you accept undergrad interns po, physical and or online internship? Thank you po.
Undergrad, yes, because here, particularly in CLSU, we have some advices. For example, we have a project, and then they come to us, and then to be part of that project. So yes, it's part of our collaboration with the universities.
Thank you, Doc. From Vay Casison of UP Diliman, how many embryos can be successfully transferred in a female carabao?
So normally, para maging safe, we just transfer a very good quality embryos. So before you transfer, nagkakaroon muna ng grading of the embryo. For example, grade A, excellent embryo, ay siguradong magde-develop ito. So therefore, kahit isa dapat kasi we are also avoiding na mag-twin kasi baka ma-endanger naman yung life ng female or cow. So, but then, kung minsan, depende sa embryo grade, baka pwede na itong B. Ganon, di ba? Baka naman dalawa. So, but then, ideally, it should be one. So, always, the excellent embryo has to be transferred.
Okay, Doc. Last question na to, Doc. Siguro,
it's okay.
Ang dami pong question kaya sobrang interested po ang mga researchers and participants natin.
Punta na lang kayo dito. May libre pa kayong pastillas, gatas.
Well, thank you, Doc.
Yes, Sir Dino. Punta kayo dito. Wait ko kayo.
Ito, Doc. What if wala pong pregnancy kit or there was no way to conduct pregnancy test? Meron po bang mga signs na pinapakita ng kalabaw that they are pregnant?
Yes, Ma'am Colette, punta kayo dito. Sagot ni Doc Alfred. Okay. Actually, even before naman na magkaroon tayo ng ultrasound, na it's now starting to be scattered or spread sa Pilipinas, ay mayroon na tayong tinatawag na yung trained yung ating mga AI technician all over the Philippines to do the manual rectal palpation. O by hand lang. But then ang ano dito, it will require mga 60 days or sometimes 90 days for us to be able to confirm the pregnancy. So what na ano pala, hindi naman pala siya pregnant, sayang naman yung 60 days na waiting. So because of that ay mapapataas pa natin, may ensure pa natin yung efficiency ng AI efficiency, and then yung reproduction rate, the reproductive efficiency simply means na yung number of calves or bulo na mapuproduce niya throughout the reproductive lifespan, bawa 15 years, sa halip na 4 or 3 lang, with this intensified, na agad-agad na detect kung hindi siya pregnant, we can produce more pregnancies or calves, sabihin natin 7, di ba? So with more calves, more milk. So that is the beauty of this intensified or yung mga advanced reproductive techniques na nagtitrain na rin kami. So yung mga dati nating AI technicians, inaano natin, na-update or ina-advance natin yung technology. So
Yes.
it's just happening now.
So, final words na po, Doc. Motivational or inspirational message po sa mga participants, researchers, na maging katulad nyo rin po.
How to be me?
Yes.
No. Katawa, ano ba? Hindi ko nga alam bakit ako naging researcher. Anyway, how do I start? You know, I was in UPLB. I started my career sa UPLB doing the cryopreservation ng buffalo sperm. So that was my MS. O kunwari, yan na rin lang yung aking yun yung trabaho ko, yun na rin yung sinideline ko sa aking pag-MS sa UPLB. And then sabi ko, pag ako ay post-doctoral study, hindi na sa semen, kundi doon naman sa oocyte para ano ba sila, aswang or complementing. So parang pinaplano mo unti-unti. And then of course, hanggang matuto ka ng reproduction and the end in mind. Na-challenge talaga ako sa buffalo since then. Kasi bakit itong buffalo na to tipid-tipid? Ba't napakahirap siyang buntisin? So ando na yung challenge from the very start. Na parang since then, alam ko naman mas mataas ang baka, no? Pero hindi ako nag-give up. So persistence and the grit, no? Perseverance. Dahil nakikita mo na yung relevance niya sa industry. So sa ngayon, habang nagiging researcher tayo, nagiging scientist tayo, the more ko nare-realize na maging relevant ka. So hindi lang sa mga awards, hindi lang sa kasi the greatest fulfillment ay yung nakaka-contribute ka, nagiging relevant ka sa organization, lalo na sa industry. So sa ngayon, dati, when I started, parang okay, hanggang research lang ako yung ganun. But then, with the turn of events, parang hindi enough na research publications. Kailangan na rin tayong I am also leading the intellectual property protection. So parang mas lumawak yung dream ko na hindi lang mag-develop, but even to commercialize. And when you commercialize, you are enhancing further its application in the industry. You are allowing yung mas maraming makagamit ng mura ng ating mga technology. So that is, I think, malapit na rin kami, no, sa na ilang years na lang. But there is the urgency for me to double time. And parang araw-araw, there is always that excitement in me na to aim high. Thinking outside the box, yung sabi ng So, this is, and of course, I'm working with my partner. So, isa rin yan sa sikreto ko. Di ba, Doc Dino? So, again, start dreaming. Sabi nga ng recently kong na-meet na Chinese na partner, oh, you have been discussing a lot of accomplishment, but I hardly hear you start dreaming. So start with a dream. And then write. And then sabi ni Secretary recently, think big, dream big, think outside the box. And then write that dream. So isulat nyo, sabi ni Boss Sanchez, isulat mo yung dream. And then act on that dream para ma-realize mo na this is it. So, sana maging inspiration yun to sa lahat. And, of course, I have 3 P's in the success, at my level now. Of course, the perseverance, the courage, the grit, talagang courage, no, amidst everything. Magpursige po tayo and focus on your dream. And of course, prayer, no? Yan, ang kailangan natin. So, I guess guys, I'm sharing not only my, but even the inner me, na sana marami tayong maging scientist na nakakatulong sa ating industry. Thank you so much.
Yes, no, napakasarap pakinggan po yung mga motivation and inspirational message po sa amin. Proceed na po tayo in our certificate. Okay, thank you po. So the Philippine Society for Developmental Biology Incorporation, o Institute of Biology, College of Science, University of the Philippines, Diliman National Complex, Diliman, Quezon City, 1101. This certificate of appreciation is given to Dr. Yofresina P. Atabay for the generous sharing of knowledge and expertise as resource speaker during the PSDB 2022 webinar series session 1 held online on April 23, 2022. Signed by John Vincent Ginabalan, Program Chair, Dr. Dino T. Tordesillas, Secretary, and Dr. Larry Ann Ipulan Colette, our President. Thank you, Dr. Atabay.
My
Congratulations.
pleasure. Hope to see you soon here at PCC.
Yes,
Maraming salamat po, Dr. Atabay. Actually, dito po ako sa Bataan ngayon. Konting ano na lang.
Daan po kayo.
Sige po.
Nice
Salamat
meeting
po.
you all.
Maraming salamat
Okay.
po, Dr. Atabay, for your talk and also for the inspirational message you gave. It's truly inspiring that you just have to take that one step forward. You have to dream, you start dreaming, and you live your dream and you write it, and you do what it takes to persevere and accomplish your goals for your dream. So maraming salamat po
Thank
doon,
you. Welcome.
Dr. Atabay. At ano po muna, picture taking po tayo ulit. We have
Okay.
our photo op number 2. So let's, for those who are still here, our 140
Oh,
participants are
thank
still
you
here.
for sticking it out with us.
So please open your camera. So we'll have our second photo session for this afternoon with our second speaker, Dr. Yofresina Atabay of the Philippine Carabao Center. So sige po, hintayin po natin ang pagbubukas ng mga camera.
Nice meeting you, Dr. Larry.
Yes po, ma'am.
Okay
Dadaan
po.
ako dyan, ma'am. Pumupunta akong CLSU eh. Daan po ako sa
Sure,
inyo.
sure. So, anuhan mo lang po si Alfred.
Ah, sige po.
Okay po.
Kung pupunta kayo dito.
Sige po, sana naka-on ang mga camera ng lahat. I have 2 panels here with me. So tingin po lahat sa camera and give your biggest smile. 1, 2, 3, smile. Okay, for the second panel. Ayan, so sige po. Baka may mga gusto pang humabol. Tingin po tayo sa... Ayan, may mga humahabol pa. Ayan po, tingin po sa camera. 1, teka lang po. May lumabas na pop-up message. 1, 2, 3, smile. Okay po. Salamat po sa lahat ng nagpa-picture. Muli po, thank you very much, Dr. Atabay, for your talk and for sharing your knowledge and inspiration to
Thank
us
you.
and to our
Welcome.
young budding scientists here in our PSDB webinar series 2022. Okay? Maraming
Thank you
salamat
so
po.
much. My privilege and pleasure. Bye.
Thank you very much, ma'am. Okay, let me just go back to my presentation. Okay, so we're done with photo session number 2. Okay, we now go to the evaluation. This is the link to our evaluation. Our PSDB board members will be posting the evaluation link in the chat box now. You may also scan the QR code you are seeing right now. Again, for you to be able to get a certificate of participation for this webinar, this session itself, kasi dalawa yung session natin. So we will issue certificates for each session. Ulitin ko po, each session. So dalawang certificate po. So isang certificate for session 1, which is today, and another certificate will be issued next week once you complete the evaluation form for next week. Okay po. So again, you must be an official member of PSDB and you should be registered in this webinar series. And you should be able to complete the evaluation form for each session, and the evaluation form, which I think is being posted right now in our chat group, the link to the evaluation form will only be open until just before midnight tonight. So hanggang 11:59 PM lang po tayo pwede fill out ng evaluation form. So please do fill out the evaluation form kung kaya na po natin. Okay? Again, maraming salamat po. Again, this is just the first, okay, of the PSDB webinar series, but the entire webinar series is presented in cooperation with Macare and together with our sponsors, Merck, MedTest, and PhilAnaserve. So these are our sponsors, and we would like to thank them very much for helping us in coming up and delivering this webinar series to our members and to other participants, and also to all interested individuals out there who want to go to the field of developmental biology here in Zoom or in YouTube, watching us over in YouTube. Okay. Wait a minute. Okay. Again, this is just the first of two webinar sessions. We still have our second session next week. Still at the same time, 2 p.m. We have Dr. Margaret Ann F. Pelayo from the NAR Institute of Science and Technology, who will talk about epigenetic mechanisms in the study of floral stem cell fate. And we also have Jobriel C. Baluyot from the Regenerative Biology Research Laboratory of the Institute of Biology from the College of Science, University of the Philippines, Diliman, who will talk about benchmarking endocrine disruptors. Okay, so so I think that is the last slide that we have for this afternoon. Let me just go back to the slide for the evaluation. Baka po